Yeah, my name is Michael Shi. On behalf of USCACA and eChina Health, welcome our annual China Pre-ASCO as summit. Yeah, so this is USCACA started our annual meeting 11 years ago in Chicago, and so we always have our members attend the meeting live. But unfortunately, since last year, we have to change the Zoom platform. And we're grateful, fortunately, to partner with eChina Health, start our virtual meeting last year. And this year, we're going to have 3 sessions. And we always appreciate the ASCO and the CSCO leadership to really, for the support of our annual meeting. And also, I would like to thank our sponsor, Junshi Biosciences, for this year's annual meeting. Welcome. So we have a great panelist and the presenter today. And first, I would like to welcome Professor Ruihua Xu, Sun Yat-sen University Cancer Center and the President-designate of Chinese Society of Clinical Oncology, gave us a welcoming remark. Welcome, Professor Xu.
Let me see. Thank you. Can you hear me?
Yes.
Okay. Dear Michael, dear Dr. Volks, dear colleagues, and all the attendees, so by the virtual, so it's my big pleasure to be invited to join this great meeting. So Pre-ASCO China Summit, so hosted by USCACA and eChina Health. So I would like to, on behalf of all the members from the CSCO, to say hello to all the colleagues. So this is a very good meeting. So actually, I personally joined the meeting in Chicago a few years ago. So as like the President-designate of the CSCO, so CSCO, it is like a very big association. We have 25,000 active members. Most of them are oncologists. So in the past 10 years, so we are doing a lot of proceeding of clinical oncology. So especially though, so we collaborate by the CSCO. So of course, we also like to collaborate with the ASCO and ESMO. So we're doing a lot of clinical practice, treat a lot of patients, but especially in the recent years, so we more emphasize in the clinical trials, especially for IND. So we like to join the global trials from the Chinese part. So we also doing a lot of IND from China pharmaceutical companies. So doing for the support of the indication in from the Chinese administration. So today, so we like to emphasize one anti-PD-1 Toripalimab in Nasopharyngeal carcinoma is one example. So we should do more. So we can contribute more for the vote, especially for the oriental cancer types like the pharyngeal carcinoma, esophageal cancer, now liver cancer. So because we have so many volume of the patients, so big the burden, so we need to do more, to find more IND or new anti-cancer drugs to contribute for the human needs. So it's my big pleasure to be here. So I very enjoy to collaborate with all the association and all the pharmaceutical company. So not only either from the global or the Chinese part. So anyway, so we are doing great jobs to get a fight for winning of the anti-cancer. So even now we are in a little bit the challenging situation for whole entire world, of course. So we are doing much better now for the situation. Anyway, thank you very much for inviting me. So thank you.
Thank you, Professor Xu. Yeah, Next, we would like to welcome Professor Everett Volks, and who is the professor at University of Chicago and our president-elect of the American Society of Clinical Oncology. Yeah, so thank you, Dr. Volk, for coming. Yeah, please give your remarks. Yeah.
Thank you very much for your kind introduction, Dr. Xu. It's a pleasure to be a part of this, and I want to welcome the members of CSCO. And of course, I represent ASCO as president-elect, with my term to start after this year's ASCO meeting. As you know, I happen to live in Chicago and work at the University of Chicago, but ASCO also happens in Chicago every year. And it's, as you know, a very big meeting with over 40,000 attendants. But unfortunately, this year still, we are going to be virtual. So we hope to see everybody, but online and virtually, and hopefully as of next year, as Chicago is opening even now, we're going to be able to welcome all of you back to a great meeting and a great city. ASCO, as you know, is dedicated to advancing cancer care. It's advancing it on a global, and we're committed to a global mission because cancer is a worldwide problem. It's not an American problem, it's a worldwide problem. And new treatments need to be made available to all patients across the globe, and of course need to be affordable so that patients need to be have access, and we want those treatments to be effective. And so that is where we see our mission in ASCO: advance the science, advance care, and do this through innovation. And it's my hope and ASCO's hope that as we all, the entire world, emerge from COVID and come back, that during these 2 years, we will have learned many new things, how to interact globally. It doesn't always have to be in person. It's wonderful if it can be in person, but it doesn't always have to be in person. But we all need to work together. We are one global community to advance the care for our patients.
Thank you, Dr. Volks.
Thank you, and thank you for inviting me.
Yeah, thank you, Dr. Volks. Thank you, Dr. Xu. Yeah, so we're always fortunate we have the CSCO and ASCO leadership to attend our annual meeting. I remember last year, Dr. Skip Burris and Professor Li Jin also attend our first session of meeting. So it's always great support from all of you. So this is a quick view of the agenda. We're gonna have 3 speakers give us very short talk, 12 minutes each. Really highlight some of the new global clinical trial progress, and then follow a panel discussion chaired by Dr. Li Yan from USCACA and the CMO of Biosciences. Okay, so again, I would like to thank our organizer and also the sponsor from Junshi Biosciences. Okay, so just a little bit technical. So we will have a Q&A session. So during the meeting, you will be able to have a chance, just follow the instruction. You select the Chinese or English, and under the button and showing a Zoom platform. And we will collect the question, and during the Q&A session, we'll be able to have you know, all the questions. So we might not be able to have all the questions, but during the presentation, during the discussion, please feel free to send a message. And also we have live broadcast streaming, and also we have a Chinese interpreter. You can choose your language if you want to have a simulcast. So thank eChina Health. We have a very great platform this year, this is a new addition compared with the last year. I hope all of you can enjoy the scientific session. All right, next slide. So it's my great pleasure to introduce our first speaker, Dr. Sheng Yao from Junshi Biosciences, and the topic of global first immune checkpoint inhibitor approved in nasopharyngeal cancer, the journal of Toripalimab. Dr. Yao. Okay, Dr. Yao, you need to present the slides from your desktop.
Dr. Yat, you are muted.
Thank you very much, Dr. Shi, for your kind introduction. I would like to thank USCACA for your invitation to attend this summit. The title of my presentation today is the Global First Immune Checkpoint Inhibitor Approval in Nasopharyngeal Carcinoma, the journey of Toripalimab. Today, I'm really honored being joined by Dr. Ruihua from Yat-sen University. The journey of Toripalimab in NPC actually was led by Professor Xu. From the phase 1 dose escalation, from the pivotal phase 2 single arm study to the global phase 3 double blind study. And without Professor Xu and all the investigators, I will not be here presenting the story today. Thank you. And here is disclaimer from the company. For those who are not familiar with Junshi Biosciences, I'm going to give a very brief introduction. Junshi was founded December 2012. Over the years, the company has established a pipeline of 30 drug candidates. We have 2 approved drugs in the market. The first one, Toripalimab, is focus of today's topic, also known as JS001, is the first Chinese domestic anti-PD-1 receive approval in December 2018. Early this year, in partnership with Eli Lilly, we have our second product in the market, Toripalimab. This is a COVID-19 neutralizing antibody, which has received emergency use authorization in the United States, Europe, and South America. Junshi has a pipeline including monoclonal antibody, bispecific antibody, antibody drug conjugates, and small molecule drugs. We have reached centers in Suzhou, Shanghai, San Francisco, California, as well as Rockville, Maryland. I'm based at Rockville, Maryland. Junshi has 2 manufacturing facilities with combined capacity of 33,000 liter capacity. And we have expansion ongoing, targeting to get a total 120,000 liter capacity with addition of 8 15,000 liter stainless steel bioreactors. Now we turn to the topic of today. Toripalimab is a novel anti-PD-1 compound. It has unique CDR sequence and a unique interaction domain on PD-1, compared with pembrolizumab and nivolumab. The crystal structure of Toripalimab in combination with PD-1 has been published in Journal of MABS. Toripalimab has a unique feature of very slow dissociation when binding to the target, which translated into a 20-fold higher affinity than pembrolizumab and about 30-fold higher affinity than nivolumab in the same assay format. In addition, Toripalimab has strong ability to induce PD-1 receptor internalization, which is independent of the ligand PD-L1 expression. We believe by removing PD-1 from surface T cell, it improves T cells' intrinsic ability in response to antigen stimulation. And in clinical study, we observed clinical efficacy of Toripalimab in both PD-L1 positive as well as PD-L1 negative cancer patients. We believe Toripalimab has a strong, unique advantage treating PD-L1 negative patients. Toripalimab currently has 18 pivotal trials. How do we choose those trials? So our strategy is not a simple repeat of what nivolumab and pembrolizumab have done in the past. First of all, we are targeting indication that has high prevalence, high incidence rate in the region. Nasopharyngeal carcinoma is a good example. In addition, we're also targeting GI cancers, gastric, esophageal, liver cancers. And we're also targeting non-small cell lung cancer patient, which harbors EGFR mutation, which has much higher prevalence in Asia than the West. From the 18 trials, we have 3 indications approved. Second-line metastatic melanoma, third-line nasopharyngeal cancer, second-line urothelial cancer. We have 3 trials have reached primary endpoint this year, including first-line NPC, first-line non-small cell lung cancer, first-line esophageal, which either have BLA file or will have BLA file shortly this year. We have additional 7 phase 3 trials will have primary readout this year. So how we started our journey in nasopharyngeal cancer. Actually, we targeted NPC very early during the development process. And it's mainly driven by clinical evidence. Toripalimab received its first IND approval December 2015. We dosed our first patient March 2016. In a dose escalation study led by Professor Xu at Yat-sen University, we dosed our first NPC patient in May 2016 during dose escalation. And we have observed a very promising early results. With that evidence, we initiated a phase 2 pivotal trial in the same year, December 2016. Here is one staining, a typical staining IHC of tumor biopsy from a nasopharyngeal cancer patient. The brown is PD-L1 staining, the blue is CD8 staining. As you can see, PD-L1 expression on NPC is, I would say, the highest among all solid tumor. Second, NPC patient tumor biopsy has very abundant infiltrating lymphocyte. This is basically a feature of a hot tumor. In addition, chronic EBV viral infection has been associated with NPC. So this naturally became a very good candidate for immunotherapy. The development of head and neck cancer by nivolumab, pembrolizumab, they all specifically excluded NPC. The reason is its unique subtype, and also the NPC patient is very limited in the West. Our phase 2 pivotal trial, Polarity-02, started December 2016. Fast forward, we received the global first approval of immune checkpoint inhibitor for treatment of metastatic NPC in February this year. The results of Polarity-02 also recently published in the Journal of Clinical Oncology, January 2021 online version. The approval by the NMPA is a conditional approval, so a confirmatory phase 3 is required. Jupiter-02 is the confirmatory phase 3 study, which is toripalimab in combination with chemotherapy for the first-line treatment of NPC. That study reached its primary endpoint September last year. A supplemental BLA has been filed to NMPA in China. And most recently, the abstract described the result of Jupiter-02 was selected by ASCO for presentation at plenary session in this year's annual meeting. We also initiated our development of toripalimab in the US in early 2018. And last year, September, toripalimab received the breakthrough designation by the US FDA for the second-line treatment of metastatic nasopharyngeal carcinoma. A BLA rolling submission was initiated this year, February 2021, to the US FDA. Here's some additional background of NPC. As mentioned, NPC is a rare cancer. Globally, incident about 1 out of 100,000, but it's endemic in southern China and Southeast Asia. You can see the incidence rate is as high as 80, so it's 80 times higher than the rest of the world. The predominant form is non-characterized NPC, which is closely associated with chronic EBV infection. And platinum-based chemotherapy is the current standard care for first-line treatment. And there is no standard second-line treatment, which represents a high unmet medical need. Both nivolumab and pembrolizumab has explored in NPC indication, and they published their findings in a relatively small cohort in 2018 and 2017, respectively. However, the development of anti-PD-1 in NPC is not all smooth sail. So recently, from Keynote 122 study, this is phase 2 study by pembrolizumab versus chemotherapy for second-line NPC. Merck recently released that it didn't meet its primary endpoint of overall survival. Polarity-02 is a study leading to the approval of toripalimab for third-line NPC. This study enrolled 190 patients from 17 participating centers in China. It's led by Professor Xu from Yat-sen University. The primary endpoint is independent review committee determine objective response rate. Here is basic patient demographics. As you can see, patients are heavily pretreated. About half the patient has received at least 2 prior lines of treatment, and 53.2% people with liver metastasis, which is a poor prognosis for NPC. Here is efficacy result from Moderator study. In the intent to treat population, the overall objective response rate is 20.5%. The response was durable. The median duration response was 12.8 months. Median overall survival is 17.4 months. Among PD-L1 positive patients, the OR is slightly higher, 27.1. However, in PD-L1 negative patient, the OR was 19.4, which is very close to the ITT population. So basically, Toripalimab works both in positive and PD-L1 negative patient population. Majority of patients were non-characterizing. We have 8 characterizing patients with a pretty good response rate of 62.5. However, the sample size is very small, so further confirmation is needed for this population. In its improved indication, 2-line plus population, the objective response rate was 23.9%. We have done extensive biomarker study in Moderator 02, and the detailed results can be found in the JCO paper. So here I'm just going to emphasize we have done a dynamic EBV, plasma EBV copy number monitoring. So what we found is early EBV titer decrease was associated with better response. At day 28 of treatment, so this is 4 weeks into treatment, before the first radiographic evaluation, for patients with 2-fold or more reduction in plasma EBV titer, the response rate is 48%. In comparison, patient without a 2-fold decrease, the response rate is only 5.7. So we believe a dynamic EBV titer monitoring can be a useful tool to predict reaction. Here is overall safety profile of Toripalimab in NPC patient. In general, Toripalimab is well tolerated with manageable safety profile. We have not observed any novel safety signals in comparison with drugs in the same class. And here is the recent regulatory milestone for Toripalimab NPC. Based on result of Moderator, Toripalimab received approval from NMPA for third-line treatment of NPC. This is a conditional approval, means a confirmatory phase 3 is required. After the breakthrough designation by US FDA last September, we started rolling submission for second-line NPC. This is for accelerated approval, also a conditional approval. A confirmatory phase 3 is required. Jupiter-02 is the confirmatory phase 3. It projects to enroll 280 patients from sites from China as well as Southeast Asia. So it's a regional, double-blind, placebo-controlled randomized trial. The leading PI is Professor Xu from Yat-sen University. In this study, we compared Toripalimab versus placebo in combination with gemcitabine plus cisplatin as first-line treatment for nasopharyngeal cancer. The primary endpoint is IRC assessed PFS. Secondary endpoints including investigator assessed PFS, OR, and OS. Recently, the result describing Jupiter-02 was selected to be presented at ASCO plenary session. And Professor Xu, we are looking forward to a presentation at ASCO. In summary, the journey of Toripalimab nasopharyngeal cancer was originated from an unmet medical need from the region, driven by strong clinical evidence during development, achieved by dedications, years of dedications by the physician, clinical staff, as well as the company. Only working together, we are very proud to provide another therapeutic option to the patient in need. With that said, I would like to acknowledge all patients participating in the trial and their families. I would like to thank all the investigators and oncologists devoted to the care of NPC patients. Thank you very much for your attention.
Thank you, Dr. Yao. Very nice presentation. And the next, so again, my name is Michael Shi. I'm the R&D head at Transcenta. So I'll share my screen. I'll be the next speaker. Okay, I hope you can see my screen here. All right, so today I'm gonna introduce some of our new development here. One of the key feature is utilizing immune tolerance breaking technology, developing therapeutic antibody. Transcenta is the company formed in 2018 by the merger of two leading biotech companies in China. One is the mAb space, have a discovery and translational research capability, with also HJB, was funded by Professor Zhao in Hangzhou. So the company, us, since the merger, we have a pretty integrated technology and also development capability in the company. So the company not only have a discovery development, also have an internal capability for manufacture using in continuous bioprocessing platform, and also proficient-based technology to develop a CMC. So the company is headquartered in Suzhou. The founder CEO is Sheng Chen, Dr. Chen. And we also have a global presence here in the United States, based in Princeton. We have the research, clinical research and the regulatory department. In China, in Beijing, Shanghai, and Guangzhou, where the major hospital and the patients are located, we have our clinical development and regulatory department. So our focus is really using technology to identify therapeutic antibody and really leverage the development pathway, initially using conducting early phase trial because the regulatory environment can start in US IND first, and then we can leverage the large patient population in China to conduct our clinical development and enroll patients really as per our development plan. We also have a technology partner, Merck KG, to developing our processing, bioprocessing, and also with the commercial collaboration, licensing opportunity with a global company like Eli Lilly. So a quick word about the immune tolerance breaking technology. So this is the antibody production platform. The technology we use is the hybridoma. It is an efficient, high developability antibody platform, but traditionally the hybridoma-based technology has a limitation because it is using the mouse developed antibodies. So for a lot, because the human protein have a lot of homology between human and rodents, so a lot of the epitope is really either hidden, also it's not immunogenic towards the mouse sequence. So the technology we use really have the capability using the peptide binding to really expose the hidden epitope and also generate the antibody for difficult to target epitope. So by screening antibody, we can screen out diverse epitope, and also you can engineer differentiate antibody. And most of the time, because all these hidden epitope and non-conserved region between human and rodents, you also identify antibody, which is easier, have a higher chance to get an IP protection. So also recently, we really leverage our continuous processing, particular perfusion-based technology, develop our CMC capability for the global clinical development, including the registration material platform to be developed. So I will go through three examples for internal development pipeline. One is the pH-dependent second-region PD-L1 antibody. Because it's the pH-related recycling, it has a local.
Hey, Dr. Shi, your audio is not very clear for some reason, at least from my end. I don't know for the other audience or panelists whether you are having the same challenges.
Okay, so can you hear me a little bit better now?
Yeah, you may want to get closer to the your microphone, etc. Try that.
All right.
Yeah, it's better. Yeah.
Okay, you think so? Yeah. Okay.
Yeah. Okay, great.
All right. Yeah. So what I show here on next slide is the one example is the MAS 2311. MSB2 2311 is a humanized antibody. This is one antibody was developed based on the unique, it is inversely related with the pH dependency. So compare with the Toripalimab, for example, these binding is really not pH dependent, but if you can see with lower pH, the binding affinity for PD-L1 is become, inversely related. So the key feature for this kind of antibody is really using the conventional antibody. Because it was once it's binding to the PD-L1 is internalized and degraded into the endosome. And for this pH recycling feature, during the transport into the early endosome to late endosome, and the antibody can dissociate in the late endosome and can be recycled out of the tumor surface, tumor cell surface, and then be reused. So this will allow the local concentration and reutilize for the to bind the target cell. So the study was conducted initially using a dose escalation study in the United States. And then we, in parallel, conducted a China study using the dose escalation and also have the expansion using biomarkers like the patient population once we reach the phase 2 dose. And on the right panels here is a combined study for the US and China, the safety profile. So the overall safety profile is very similar to the marketed PD-L1 inhibitor, for example, the Toripalimab. This is the preliminary efficacy data we developed based on the China study. And this is, as I mentioned, this is a biomarker selected patient population using PD-L1, MSI high, or EBV positive tumor type. And so when we have the expansion phase, which is 20 milligram per kilogram Q3 week or 10 milligram per Q2 week, the study was expanded in this biomarker selected patient. Preliminary data show in the expansion phase, the tumor shrinkage was observed. 44% of the patient achieve a partial response in the 20 milligram per kilogram dose per Q3 week. And also one patient was observed with a PR in the 10 milligram per Q2 week dose. There's also additional one patient have irises response also observed. So this is the data cutoff for June last year. And later at the upcoming ASCO, we're going to have a later data cutoff to show the additional data for this clinical trial. And also among these expansion phase, we observed 3 out of the TMB high, 8 TMB high patients achieve a partial response. As you know, Pembrolizumab was approved in the TMB high patient population by the US FDA. So we are currently continuing the expansion in TMB high patient population, further investigate the clinical activity in this tumor type. So this is a first and only pH-dependent recycling PD-L1 antibody. We are continue to develop it in further expand other single agent or combination with other target therapy. The other example I want to show here is a Claudin 18.2. This is a tight junction protein, which is embedded in the transmembrane protein, and there's very limited sequence exposed to the extracellular part. Traditionally, it makes it a very hard target for antibody production. Claudin 18.2 is highly expressed in the GI-related tumor type, for example, gastric cancer, pancreatic cancer, and esophageal cancer, biliary tract cancer. So this is a really, according to Dr. Xu's opening remarks, right? This is a highly prevalent Asian cancer type, which is has a huge unmet need. So the Astellas, through the acquisition of Ganimab, they acquired IMAB 362 was the first in-class molecule targeting Claudin 18.2. The FAST data was reported earlier this year, really established clinical proof of concept, and we are continuing to support, pursue this gastric cancer development, other GI tumor development. The mechanism for this Claudin 18.2 antibody is really trigger the ADCC-CDC activity and leading to the tumor killing. So we are interested to looking the expression of Claudin 18.2 because almost half of the patient in gastric cancer express medium to high Claudin 18.2, and the zolbetuximab, the activity for FAST2 only show there's about a very high expression of the Claudin 18.2. Those patients with over 75% IHC positive can show the clinical activity. So what's the difference between TST-001 and zolbetuximab? It binds to different epitope and also have a higher affinity compared with the Zobi. And another feature is we have engineered with a reduced Fc receptor and with the enhanced Fc receptor binding of the NK cell to leading to the tumor killing. So the affinity of the TST-001 with a target Claudin 18.2 is about 10 times more potent compared with the Zobi. And also because the reduced fructose and enhanced ADCC activity, the ADCC killing will actually could be 10 to 100-fold higher, more potent in Claudin 18.2 relative low expressed gastric cancer cells. And also on the right-hand panel here, we can see the tumor express anti-tumor activity of Zobi in the median high Claudin 18.2 expressed tumor type. And at the 10 milligram per kilogram, we actually observed 7 of the 10 animal with the tumor complete disappearance, right? And none of the IMAB control group showed the complete tumor shrinkage. So in parallel, as we develop a target therapy, we also realized that antibody detection, right, for the biomarker selection is also in parallel is important because what we've observed is traditional Claudin 18.2 antibody detection is not very specific. Even with the Zobi selected antibody, we've observed it is actually cross-reactive Claudin 18.1. So in parallel, we develop another diagnosis antibody to more precisely select the Claudin 18.2 tumor type. And this will allow for have a more precise selection patient down the road to have a broader patient population selection. Last ESMO, we have heard exciting news about the CheckMate 649 results, right? Nivolumab combined with chemotherapy in first-line cancer type. really show improved survival. What the report is really CSP higher than 1 patient have, that's the major driving force. Low PD-L1 expressed, the 2 patient response is very similar to the chemo. And we also collaborate with academic researcher. We have observed Claudin 18.2 overexpressed patient, usually not co-expressed with PD-L1 or HER2. And so this is a patient population, sub-patient population, really targeting Claudin 18.2 can have a more precise, more targeted approach, enhance the tumor killing. So by looking at all these Claudin 18 expressed the tumor type, on the right upper panel here, we can see combination with chemotherapy, both the CAPOS in first line or the paclitaxel in PDX model show added effect of TST001 in combination with chemotherapy. In addition, in the gastric cancer, we know the VEGF inhibitor like the ramucirumab also is proved in the second line setting. TST001 in combination with VEGF inhibitor in this case is a mouse analog DC101, which is a monoclonal antibody against the VEGF receptor R2, also see synergistic activity. In addition, on the lower right panel here, you can see combination with the PD-L1 inhibitor, atezolizumab, also show in the xenograft model have added synergistic anti-tumor activity. So in patients with both Claudin 18 or PD-L1 expression, but not the most significant expressed patient, the combination approach will have added effect. So this is an opportunity for us to further explore the combination study. The TST001 was investigated in both the China and the US. So there are 2 studies are currently ongoing. We are still in the dose escalation phase, but once we reach the phase 2 dose, we can expand in multiple tumor type with the Claudin 18 overexpressed patient. That include the gastric cancer and also non-gastric cancer, for example, other GI-related tumor type like pancreatic cancer or biliary tract cancer. So this is currently will be in the expansion phase in the next, in later half of the year. And in addition, in the China study, we also started the combination therapy, combination with the first line ZLOG, CAPOX, and also the second line with paclitaxel. Both dose escalation phase with chemo already started. We actually started dosing the patient just recently. So with the development, we are interested to really looking at the landscape of a current development, really thinking about both the first line and also the potential future landscape for chemotherapy and the immunotherapy failure patient. There will be a lot of patients with the Claudin 18.2 overexpression, which is represented about half of the patient, which is really have a great opportunity for us to develop this targeted therapy. And the last example I want to just highlight, there's a bifunctional molecule, the TST005. It's a next generation PD-L1 TGF-beta trap bifunctional molecule. It has a the PD-L1 binding arm, which have a high potency versus binding PD-L1, and what also have a TGF-beta R2 trap fused with the Fc region. And what is really unique for this PD-L1 arm is there has a Fc silencing feature, which will allow to prevent ADCC activity. That way, because the current molecule, as you observe, is the M7824, the Merck KG molecule, bifunctional molecule. They recently showed they failed frontline head-to-head comparison with the Keytruda in non-small cell lung cancer in PD-L1 high patient, which is we think there are probably two reasons. One is this FC region is the backbone is avelumab, which is also has potential to killing the effector cells through ADCC activity. And the other one, which is really important in consideration for clinical development, if they have not yet enriched the TGF-beta selection, because TGF-beta cause immune exclusion, right? It has prevent the lymphocyte infiltration. So it's this very important target. If we can select those patients, that will allow to develop a more effective treatment. So in this right-hand center, we can compare with the PD-1 and TST005 have enhanced T cell activation activity. And also on the lower panel, we can see the binding of a PD-L1 to active the T cells is really efficient, comparable with PD-L1 binding. And also more importantly, because the mutated FC receptor binding region, it completely eliminated ADCC and CCD activity, which have the not only killing the tumor cell, but also have the capability to killing some of the T cell effector cell. By eliminating this FC region, they will completely abolish this ADCC activity, which have opportunity to differentiate from the existing current molecule. The other feature is also they have enhanced the TGF-beta stable trapping. We observed the M7824 because they have an intact receptor TGF-beta R2 fusion region. They can be exposed with the enzyme cleavage. So for the Western blot, you can see the TGF-beta trap is not very stable. On the other hand, TST001 express the cell line, we have pretty stable TGF-beta trap. And translate into the preclinical data, we can see the exposure of the PK-PD relationship. You have enhanced the PK exposure because the stable trap, and also have a sustained the TGF-beta trapping activity during the 3 weeks dosing. You can see the inhibition of the TGF-beta and the PK profile also sustained. And on the right-hand panel here, we can see the downstream target inhibition of PSMAD continue to be inhibited by TST005 in the in vivo study. And also, in addition, one important feature, right, we mentioned about the TGF-beta is the immune exclusion. In tumors with who are sensitive to PD-1, PD-L1 binding, we can see the TST005 have a potent activity against cause tumor shrinkage. On the left-hand lower hand figure here, and in the middle figure, we can also see that correspond to the CD8 and the CD45 really enhance the T cell infiltration into the tumors. And what is really important for this, what I mentioned about the tumor biomarker selection, in TGF-beta enriched tumor model here is the EMT6 model on the right-hand panel here. Traditionally, is not the immune responsive tumor type. PD-1 has a very limited, anti-PD-1 has a very limited activity due to high level expression TGF-beta. So the using the TST005, we were able to inhibit very potently in this immune non-responsive tumor type. So the clinical development is started now. We just retained the US IND. We're starting this first in human started in China, in the US, and we using the same protocol. The China and the US started this phase 1 proof of concept trial in both US and China using the same protocol. As we mentioned before, the US regulator, we can have the IND clearance ahead of China. So by the time when the Chinese center can join the study, they will be able to start at a higher dose. That's shorten our development timeline. So I think next year we're going to have more data to report. And with that, I want to thank all the organizer for the invitation. And really, through the collaboration, we really want to develop innovative medicine to benefit the future patients around the world. So with that, I will stop here and introduce Dr. David Fogelman from Merck to get the next talk.
Okay, thank you, Dr. Shi. I appreciate the opportunity to present. Let me introduce you to my 5-year-old.
No shower tonight.
So, okay, I'll be out in a minute. Thank you.
No shower tonight.
He doesn't want to shower tonight. Okay, you win. No shower tonight. Okay. Anyway, so with that introduction from my 5-year-old, let me go ahead and share my screen. Okay, you gotta go. Bye. Okay, I wasn't expecting that. Anyway, nice to meet you. I'm Dr. Fogelman. I'm a medical oncologist. I trained at the Albert Einstein College of Medicine. I've spent 13 years on the faculty of MD Anderson, where I treated patients with colorectal and pancreatic cancers. And most recently, I've moved to Merck, where I have the privilege of helping design and run clinical trials in colorectal cancer. So again, I'm a Merck employee, that's my disclosure. And my goals for you today are to review some of the recent targeted data on noteworthy colorectal cancer targets. And I will, in the process, try to highlight some of the upcoming colorectal cancer abstracts at ASCO 2021. Now, obviously, a talk like this could theoretically involve a great many targets. While I'd love to teach a course on it, for the purpose of tonight, let's focus on 3: HER2, KRAS, and VEGF. So by way of background, HER2 overexpression is noted in 2% to 5% of patients with colorectal cancer. Overexpression is more frequent in KRAS and BRAF wild type patients. In stage 4, roughly 5.2% of wild type patients as compared to 1% in mutated patients. And likewise in stages 2 to 3, 2.1% in wild type patients as compared to less than 1% in mutated patients. Activation of the HER2 pathway in turn activates RAS and PI3 kinase. And this may be a mechanism of cetuximab resistance in patients who are otherwise KRAS wild type. So moving on to some of the more recent clinical trials. The trastuzumab and lapatinib study looked at 948 patients that they screened. This is the Heracles study for HER2 positivity. After the screening, they came up with 27 patients that they were able to enroll in the clinical trial. And of these, now that said, of these 27, as you see, response rates were actually pretty good, 30% for a heavily pretreated population, and clinical benefit was seen in 74%. Overall survival was 46 weeks, and toxicity was mild with grade 3 events, including fatigue, rash, and bilirubin. Now, one important lesson from the study is that patients with greater overexpression actually seem to do better than patients with less overexpression. And you can see that on the Kaplan-Meier curve of progression-free survival here, where benefit appears to correlate with HER2 copy number. And thinking about this in terms of the upcoming ASCO meeting, I'd encourage you to look at a trial in progress, which is now going on with trastuzumab and tucatinib. Tucatinib is slightly different from lapatinib. Tucatinib targets HER2 and HER3, where lapatinib targets HER1 and HER2, but I'll personally be very interested to see the outcome of that study. So moving on, another drug that's been used is pertuzumab in combination with trastuzumab. Now, this was a basket study at multiple centers led by one of my former colleagues, Funda Merritt-Bernstein at MD Anderson. And in this case, they looked at a group of 57 patients. They found a response rate of 32%, clinical benefit of 44%. But I think the lesson to take from this study is that unlike the former study, this study actually did look at KRAS wild type and KRAS mutant patients. The wild type patients, 17 out of 43 responded, but of the mutant patients, only 1 out of 13 responded. I think that's an important lesson. And just so you know, that this study will be updated at ASCO, and there's a number of related abstracts that I think will be worth your while to look at the upcoming meeting. Another drug that's been used is TDM1. TDM1 is trastuzumab linked to DM1 is it's a microtubule inhibitor, more potent than vincristine. And this was actually the second cohort of the Heracles study. This was the same group that did lapatinib. And what's important in this study is that they assigned patients, they stratified based on IHC overexpression. And what they observed is that if you're in that higher IHC group, your PFS is going to be much longer than if you're in the lower IHC 2+ group. And finally, trastuzumab deruxtecan. So this is a topoisomerase inhibitor that's been approved in breast cancer, and it's also approved in gastric cancer, and of course now being tested in colorectal. And again, when you look at that IHC 3+ or the IHC 2+ with FISH positivity group, you get a nice response rate, 45%, and a PFS of 6.9 months, which appears to be much better than cohorts B and C, where they took patients with less overexpression. And when you have less overexpression, your PFS is much shorter, 1.5 months. So it appears that the final results of the study will be presented at ASCO. And again, I'm very excited to see the results of that. So just to sum up HER2 disease, again, these are a comparatively uncommon group of cancers, but they do appear, at least if you are KRAS wild type, to be highly druggable. And likewise, the degree of overexpression does seem to correlate with your degree of efficacy. And this, again, happily, this will be covered in fair detail at the ASCO meeting. It would be remiss of me not to point out KRAS mutation as a prevalent barrier to CRC treatment. This is highly prevalent, over 40%, and if you include NRAS and HRAS, closer to 50. It's associated with resistance to EGFR therapy, and as well as resistance to HER2 therapy, and it leads to downstream activation of the MAP kinase pathway, BRAF, MEK, and other proteins. Now, we've been trying to deal with this for decades. Initial efforts looked at farnesyltransferase inhibitors, which did not work because they failed to stop an alternative processing via GGTase. Unfortunately, when you try to target GGTase and farnesyltransferase, the results are highly toxic. And likewise, earlier efforts have tried to look at RAS trafficking. Two drugs, salarasib and deltaracin, have unfortunately both been, well, they're not in clinical development anymore. And my understanding is that those were also fairly toxic drugs. So more recent efforts are now looking at ways to allosterically inhibit particular variants of RAS mutations. AMG 510 and Mirati's 849 are two examples of drugs that are specifically targeting KRAS G12C. The AMG 510, and just by way of disclosure, I contributed patients to the study when I was on faculty at Anderson. But that said, the AMG 510 is probably furthest along. And what we see is that, unfortunately, although you see a fair response rate in non-small cell lung cancer, 48%, the response rate in colorectal cancer is much less, 3.4%. That said, you do see a reasonable incidence of at least disease stability. And so it's not clear why there's such a big difference between diseases. Questions that arise are whether or not this is something related to the innate biology of the tumor cells, or alternatively, perhaps it's something related to the immune microenvironment. And really, we one need that we have for KRAS mutation is to understand mechanisms of resistance to those direct inhibitors. There will be, in fact, an upcoming ASCO presentation. This is abstract 3127, looking at the landscape of KRAS G12C and associated genomic alterations and interrelation with immuno-oncology. That should be a great talk. And full disclosure, I have no idea what they're going to say. Finally, let's look at the Vegf pathway. Now, of course, bevacizumab has long been established as an effective treatment, if imperfectly so, for colorectal cancer. Some recent events have included the TASCO-1 study, which looked at combining bevacizumab with TAS-102. This was done in patients who could not tolerate more aggressive treatment. They did see when compared to capecitabine and bevacizumab, they did see a somewhat longer, but non-statistically longer increase in PFS. There will be more mature data, I hope, coming from the similar study. This is the TRUSTY study looking at the same combination in second-line colorectal cancer. That's going to be presented at ASCO this year. Likewise, a study of ramucirumab in combination with TAS-102 will be discussed at this year's ASCO meeting. And finally, just looking at moving from bevacizumab as a relatively clean VEGF agent to some of the multi-targeted molecules. Again, this is a Merck product. This is lenvatinib. So lenvatinib inhibits both VEGF and FGFR. At the GI ASCO meeting earlier this year, Dr. Gomez-Roca presented the LEAP-005 study in MSS colorectal cancer patients, finding a response rate of 22% and a disease control rate of 47%. Now, this is something that might be compared to the Rego-Nivo data, which has already been published in early form. But just to show you what's coming up at ASCO, it looks like we will be seeing an update of the Rego-Nivo study. And so I think, again, I think there is room for the multi-targeted VEGF inhibitors in combination with other drugs. IO therapy has been one combination. And again, we'll see the data at this year's ASCO meeting from this study. And that said, thank you very much for your attention.
Yeah, thank you, Dr. Fogelman. Yeah, so yeah, we are running a little bit behind, but let me introduce Dr. Lian and the panelists. And Dr. Lian is the managing director of USCACA, and is really the driving force behind our organization. And he's the CMO of BreeBio. And Dr. Yan.
All right, great. Thank you, all the speakers, and really great talks. David, you gave 28 slides presentation in less than 10 minutes. And just out of curiosity, I actually listened to the real-time translation in Chinese. The guy actually caught up with your pace, so that was pretty amazing.
Oh, that's what you get for growing up in New York City. You have to talk
That's right,
fast.
yeah. So let's just get started. I didn't see any questions from the audience, so I do have some questions for each speaker. Let's start with Dr. Yao. So, first, I just want to congratulate on your collaboration with Professor Xu, getting the really outstanding data in NPC for your anti-PD-1 antibodies. And very happy to see that you are actually getting the drug to the US through the BLA application process. Two questions. One is, are you also planning of registering your drug with other indications in the US? And the second question is actually for Dr. Volks, in terms of what are your thoughts with data generated from another country overseas, and then once they got approved by the US FDA in terms of prescribing oncologists, your view in terms of using them in your clinical practice?
Thank you, Dr. Yan. So in my company overview slide, I did mention, I don't have to talk about it in detail. So we have recently partnered with Coherus Biosciences for North America commercialization. So we are going to work with Coherus to file multiple indications, including NPC. And also our partner Coherus also announced that we do plan to file large indications such as non-small cell lung cancer. So we're going to evaluate the totality of the package, the trials, and we're going to communicate with regulatory authority here in the US and to see if we can get any additional phase 3 result approved here in the US.
Thank you.
So if I could answer your question, first of all, my compliments. This is a wonderful trial. It's a wonderful endeavor. This is a trial that could probably only be done in China because the frequency and the incidence of nasopharyngeal cancer is higher. And so to really advance care for those patients is wonderful. And I look forward to the presentation at ASCO. We know in the United States that drugs like pembrolizumab do have some activity in nasopharyngeal cancer. I myself have a busy head and neck cancer program, although nasopharynx cancer is a minority of that for us, but we see some, and we use available drugs. I think if a evidence-based drug is approved, and your drug would be evidence-based, presumably based on a phase 3 trial, then I think that that is the drug we would use. And the fact that the data were generated in another country, that's common these days. And so I don't think that that's a handicap. I can't speak for the FDA. I don't know how they would look at that. But certainly as practicing doctors, we would be very open-minded and be guided by the data.
Great, thank you so much. Dr. Volks, maybe I'll just piggyback on your answer about this NPC question. We know there are numerous anti-PD-1, PD-L1 either being marketed or still being developed, as highlighted by the 2 talks that we heard today. From your perspective, how many more PD-1, PD-L1 antibodies you would need in the clinical practice in the US? Well, we're saturated.
At some point, what I would like to see, but this is a problem in the United States, is that companies would start to compete on price because the activity of the drug seems to be similar. And what is also similar is the price of the drugs. And, at some point, I would hope that one of the companies would say, well, maybe we can charge a little bit less and, have in return, maybe easier access for the patients to the drug. So we don't need more drugs that target the same target. And, that doesn't mean that the drugs that come on later are better or worse. And trying to find new indications is very worthwhile. And nasopharyngeal cancer is certainly a new indication. That's terrific. But I would much rather see companies focus on new things, on new targets and advanced science, rather than just going for what is really in the end marketing and a commercial gain. It's not a scientific gain.
Absolutely. Just some comments in terms of the pricing of anti-PD-1, PD-L1 antibodies in China. I know Dr. Yao probably lived through that, because I believe there are at least 6 marketed antibodies in China in the market. The price, the good news, I mean, for patient perspective, the price in China is only a fraction of that in the US. But of course, the bad thing for the sponsors of the companies is a lot of R&D investment. They may or may not be able to even recover all the investment. So that will hinder the additional future R&D efforts. But that's a bigger topic for another day. I just got a new update from our broadcasting agency that there are 8,300 people watching live streaming as we speak. And I just saw we have a couple questions from the audience that just came out. Okay, so Dr. Yao, there's a question for you. I believe it says for your Toripalimab, but do you foresee that it will be effective in other EBV-related malignancies such as Burkitt's lymphoma, for example?
Right. So I just typed in the answer. I think in general, as viral-induced tumor are more immunogenic and have higher chance to respond to immunotherapy.
Okay, great. Yeah. Thank you so much.
Maybe I can do some comment on the question. EB was especially so related to different type of cancer to gastric cancer. 10% of the EB virus associated. But unfortunately, from the first finding from the Korea colleagues, so the report in Nature Medicine, I remember maybe in a group, so 6 or 7 patients with EB virus positive gastric cancer get all patient response. But unfortunately, from our data, so this is the prospective trials for single arm phase 2. So it cannot be repeated. So the response rate is quite similar with the other the non-EB virus gastric cancer. That's a finding.
Okay, great. Thank you, Professor Xu, for your added comments. Really appreciate it. So Michael Shi, as we were discussing in terms of innovation in anti-PD-1, PD-L1, I mean, apparently you have a very interesting candidate, anti-PD-1, PD-L1, which is PhD dependent recycling features, very unique. From your perspective, and I also would like to hear from Dr. Volks and David from your oncologist perspective, what kind of additional gain in terms of efficacy that you think will gain traction in terms of not only to get registered, but also to get adopted in clinical practice? I think that's going to guide your clinical development.
I think that's good question, right? So this is something where we observe, earlier efficacy is really encouraging, right? If you have a biomarker pre-selections are over 40%, 50%, it's actually quite encouraging. And again, right, this is something what we say is really each molecule has a unique feature. If we have appropriate biomarker guided selection, truly leverage some of these clinical activities, it's really in the end, along with the the manufacturing, the lower cost, also addressing Dr. Volk's question, could potentially benefit not only from the clinical efficacy perspective, but also this perfusion-based manufacturing can tremendously lower the cost, will eventually benefit the patients. So this is a combination of clinical development strategy, but also some of the manufacturing innovation, I think will really drive down the cost, and also along with other combination approach, we hope can really encourage increase of the efficacy. Yeah. Dr.
Yeah, so from my perspective, this was a really interesting presentation, Dr. Shi, and I appreciate that. I can't really speak to the relevance of pH dependence. I think that will be in the data in the end that we'll need to see. The other compound you presented I thought was very interesting is the PD-L1 TGF-beta trap, and of course we are investigating currently bintrafusp, which is a earlier compound. And it would be interesting to see the concept of combining and targeting both of these molecules is very, very interesting. And I look forward to seeing clinical data.
Yeah, thank you, Dr. Volks. Yeah, we're also very excited, right? From the design perspective, we put in a lot of thoughts behind that, really looking for how to differentiate with the existing molecule, even though we're slightly behind. But I think as I mentioned, the bintrafusp, kind of my suspicion is really twofold. One is the PD-L1 arm, right? Intrinsically, avelumab, it seems to be a weaker PD-L1 inhibitor. And also, really from our perspective, really you should leverage the TGF-beta pathway by really looking at translational research, biomarker patient selection, and really identify these opportunity to identify these patients who are resistant to turn the cold tumor into hot tumor. That's where this compound has a potential. So certainly we'll be interested to see next ASCO or so we have some preliminary data to show. Yeah.
Yeah, I look forward to that. And do you have any biomarkers for the TGF-beta compound?
Yeah, so we are looking quite a few these biomarker. You know, the early evidence, as you can see, the Genentech has done the atezolizumab in bladder cancer. There's a TGF-beta panel, right? They tested. That's really leading to some of this investigation. So we're certainly heavily investing in this translation research to really combine the molecular feature of these engineered product and also with the companion diagnosis to select the patient who are likely to respond.
That's terrific.
That's really good. Michael, you got a question from the audience. I don't know if you want to answer that question live or you want to type your answer. But in the meantime, as we're seeing that there are a lot of innovations happening globally, not only in the US, but also in China. Dr. Volks, as an ASCO president-elect, could you comment in terms of how ASCO as the largest clinical oncologist organization to really promote accommodate as well as promote early phase clinical trial investigation of new agents coming from China, for example, to really harmonize the development between these two countries?
Yeah, I think ASCO takes a responsibility of really being a global organization. And we have a responsibility to patients worldwide. Our constituents are doctors and patients. And of course, we want high quality care, good care, innovative care globally. And throughout our history at ASCO, these have been international meetings. And what I've observed, I've been a member for many years, is that over the years, Europe became more important. And that was maybe in the 90s, in the 80s and 90s. And in the last 20 years, Asia, and in the last 10, 20 years, China in particular, has become a very, very important partner and a very important component. And you see that by the number of abstracts at ASCO meetings and at the associated meetings that ASCO does. So there's very nice interaction with CSCO. And I would hope that during my presidency, we can enhance that, hopefully in person. But certainly I see us as partners, and I would hope we will work very closely together.
Yeah, thank you so much. I would also like to ask Professor Xu, as a president-elect for the largest clinical oncologist organization globally. What are your thoughts in terms of working with innovative companies to really promote, especially early phase biomarker-driven studies, so that we're not repeating, just running a lot of phase 3 of the same agents against the same targets?
Yeah, and so it's a good question. So I very appreciate Dr. Volks' the comments. So doing that collaboration in the future to make more contribution from China part, so oncologists to take care of the patient. So you mentioned, so I didn't catch up your question well because the internet problem. So we may not to take a lot of effort only in the a phase 3 trial, that big trial. So we should pay more attention to biomarker driven early develop. So I totally agree. So right now, so we do not have enough clinical cost to that. So and for the phase 3 trials. So I think sometimes that we should do more, like the early phase with the target driven or biomarker driven trials to get more that the anti-cancer effect go to the patient at the best side. So right now, as maybe we are doing too much for that, only the phase 3 confirmation. So we do not have that enough resource right now. So I feel very stressful in clinic. So we take a lot of clinical trials because we are the biggest one in China, so the cancer center. So So many stressful. To try to find a PI, it's very tough. Right now. Thank you.
Yeah. Great, thank you so much. So maybe a proposal for both ASCO and the CSCO, maybe we could, promote more innovative early phase trials rather than to occupy all the investigator and the patient resources with a large phase 3, but relatively redundant, investigations. So that might be a good topic to discuss at the future ASCO or CSCO meetings. With that, I think, the discussion has been really outstanding, but I noticed that we're 20 minutes post the time mark. I just want to invite, our presidents and the speakers to see if you have any final thoughts and comments before I close this wonderful, wonderful session.
Thank you.
Yeah. That's a great session. Yeah, so I think in the interest of time, we'll probably just wrap it up. Yeah, and we have one more slide and we can present. So on behalf of USCACA and eChina Health, I again thank all the speakers and panelists for this great discussion. And as usual, we tried a lot of interest, and I'm so pleased to see we have over 8,300 people actually looking at our stream, live stream. Just a quick note, right? This is our first session. I really want to thank Dr. Volks and Dr. Xu representing the ASCO and CSCO to really, kick off this session. So on May 22nd, we have another session and China Innovation Drug Licensing to Global Pharma. And May 29th, right before ASCO, we have a third session. Is the ADC drugs international clinical research and the progress. That's actually a live meeting with, so there will be, I think you're registered, it should be okay. So just go ahead and attend that 2 sessions. And so I would again like to thank Junshi for providing funding to support. And as usual, we have a great success for our annual meeting. I really thank the speakers and panelists and also the attendees, right? Sacrificing your late night in the weekend and early morning in the weekend to stay with us. It generated a lot of interest. I hope to see you in the next session. Yeah, thank you very much.
Thank you very much.
All right, thank you.
Thank you, everyone.
Bye-bye.
Bye-bye.
Bye.
Bye.
Thank you. Bye-bye.
Thank you.